Welcome, biologists, to this screencast covering Chapter 3, moving us into enzymes. Now, hopefully you've already had a bit of a look at this and have started considering it in preparation for your poster work. And also, hopefully you've had a good break and have enjoyed the holidays a fair bit and are ready to hit the ground running and start consolidating the reading and the work that you have done, and start building on that in preparation to keep moving forward. So we've got plenty of time to learn the work, review the work, consolidate it, test it, and apply it before we get to the real assessment stuff, which is obviously going to be the nitty-gritty. So moving into Chapter 3A, we're going to start learning about what enzymes are. That's how we're going to kick things off. So the learning intention is to learn what enzymes are, about their structure and their function. And so by the end of this, you should be able to define enzymes and explain the structure and function of enzymes, the importance of their three-dimensional shape, and the structures within those three-dimensional shapes. So we'll walk you through that. Now, this is in part for review before class. So by the end of Chapter 3A and 3B, so the entirety of Chapter 3, you should be able to define the enzyme, what they do, and their key features, identify and explain the key factors that alter enzyme activity. And we looked at one of those in the practical that you did late last year and that you're meant to be writing your poster on, and the role of enzymes and coenzymes in biochemical pathways. And that becomes particularly important as we move on to chapters 4 and 5, and we explore photosynthesis and cellular respiration in more detail than you did in year 11. The purpose of this screencast is to support your reading, your personal revision, and your application of this work in preparation of your poster. So in class, I intend to recap this with a modified version, but you're going to provide the key information. We're going to build notes together, and you're going to demonstrate your understanding. So as you watch this screencast, as you do your reading, as you take your notes, as you have a go at the questions in the chapters of your textbook or online with Ed Rollo, please, if you get stuck or you're unsure, ask me questions, get assistance, get things clarified as you work. And then obviously at the end, if it's still not clear or you've forgotten or something else has come up and you're not sure about it, ask again at the end, at any time, right through till November. Make sure that we keep reviewing this and improving it. But let's get into it. So what is an enzyme? Basically, it's a biological or carbon-based catalyst. So there are catalysts that are not enzymes, but all enzymes are in fact catalysts, if that makes sense. So there are other substances that can catalyze reactions that are not considered an enzyme, because to be an enzyme, they must be biological, carbon-based, and so there is a key difference. But all enzymes are in fact catalysts, and any catalyst, regardless of whether it's an enzyme or not, speeds up chemical reactions. In terms of enzymes, they are almost universally, without exception, proteins, and that's going to build on the learning you've done around nucleic acids and proteins in the work last year. So they are protein structures. This is from your textbook on page 129. I'm not going to actually answer the question, you are, but I want you to look at this image here, which is a picture of maltase, which is an enzyme that breaks down the sugar maltose. But when you look at this structure, what have you learnt about that you can see in this structure? I'm going to ask you that question when we return to class, and you're going to be able to answer it. But over here, this is more important. This is the more common representation you're going to see of enzymes. It also helps explain the background image that I've used, because enzymes typically look a bit like Pac-Man. But here you've got your enzyme, the maltase. Maltose is what we call the substrate. Enzymes act on a substrate and either break them apart or stick them together. We'll go through that in a moment. And then another key part of this structure is this three-dimensional shape that's formed into what we call the active site. We'll go through that and talk about that as we go. So the features of enzymes on page 129, I'm not going to go through this on this slide. I'm going to go through each of these individually. Generally speaking, it'll be very briefly. You can read what each of the boxes say, but it does say this is figure 2 from page 129. These are the key features of enzymes that says you should know. You actually must know them. All right, it's really important. So let's go through them one at a time. Enzymes are reusable. They bind to substrate. They activate the chemical reaction. They release the substrate, and they are unchanged, meaning that they can bind to another lot of substrate, make that go through a chemical reaction, and release and repeat that over and over and over again. So they complete or catalyze the chemical reaction multiple times. One enzyme can actually drive multiple chemical reactions with the same substrate. All right, so that's important. They're reusable. They are highly specific. Their active site is a very specific shape and will only bind to very specific substrates. So that maltase can't bind to lactose and also break down lactose sugar. It can only act on maltose because it's highly specific. So that, again, really important. Most of the enzymes that are catalyzed by enzymes are reversible. And so you'll see, and we're going to learn these terms as we go through, if they build up larger molecules, it's referred to as an anabolic enzymatic chemical reaction. If they break them down, so they take the substrate and break it down, it's referred to as catabolic. So anabolic and catabolic, we'll learn about that some more as well. Tipping Zach is already all over that because they are commonly used terms in gyms for a different reason. So we're still working our way through. They speed up chemical reactions, but they are not involved in creating products. Right? So the chemical reaction is the chemical reaction. All they do is make them go faster. So you have literally billions upon billions upon billions of chemical reactions happen in your body every single second of every single day. If you did not have enzymes, those reactions would all slow down to the point where you can't survive. It's actually the principle behind hypothermia, you might recall. You get cold, the enzymes work slower. If the chemical reactions happen slower because the enzymes are working slower, all the chemical reactions slow down, you die. These chemical reactions would happen if you allowed enough time. They still will happen, they just won't happen fast enough. Enzymes speed them up so they do happen fast enough. It's why they're important for all living things, but they're important to making sure chemical reactions happen in the time frame that is required. So they make them go faster, but they are not responsible in any way for making the product. So I've already touched on the active site. It is a very specific area that binds the substrate and leads to the reaction occurring. That's all I'm going to say about that. I've already mentioned this, that they're generally speaking proteins. That's really all you need to know there. I've already also mentioned that enzymes are all considered catalysts, but there are other catalysts that are not considered enzymes. So that's fairly intuitive, and I hope you understand that. We're going to look at this in more detail, particularly with respect to photosynthesis and cellular respiration, but they act on entire biochemical pathways. By a biochemical pathway, if you think about cellular respiration, glucose comes in as a substrate. It goes through a number of steps, right? And you know some of those steps. It goes through glycolysis and the Krebs cycle, the electron transport chain, but it goes through a whole bunch of steps to generate the production of ATP. But that entire pathway going from glucose to ATP at the end is referred to as a biochemical pathway, and there are enzymes at every single step along the way, catalyzing those chemical reactions and driving that process on. Most enzymes end in A's, that's all I'm going to say. And when you represent a chemical reaction that involves enzymes, you will write the enzyme name above the arrow, and I'll show you an example of that in just a moment. So on page 130, it goes into what is actually going on with an enzyme catalyzed reaction. So basically, you've got your enzyme with its active site exposed. The substrate comes in and binds to that active site. Now, typically what an enzyme does, it'll slightly alter the position of the substrate or just twist it in such a way that the bonds that are holding the substrate together are more easily broken. In other words, it makes it easier for a chemical reaction to start, because chemical reactions are taking a whole bunch of chemicals, busting the chemical bonds, and then sticking them back together in a different configuration. So products come in and reactants come in and different products come out. All the enzyme does is just make it easier for that to happen, and I'll touch on the specifics of that in just a moment. But it binds, and it will lead to a change in shape of the active site binding the substrate. It will lead to the reaction occurring, which changes the chemical composition of the substrate. So what goes in is not the same as what comes out. And then it releases those again. And again, importantly, you can see the active site is left unaltered from the beginning. So it's unaltered, meaning that more of this substrate can come in and the process can loop around over and over and over again. Now, on that, it talked about slightly changing the shape of the active site. There's two theories with respect to how an enzyme binds specifically to its substrate at its active site. So there's two key theories, and the title of this slide tells you what they are: induced fit versus a lock and key. We've already learnt that the shape of all proteins is really important, but hopefully it's already really clear to you that the shape of enzymes, and in particular their active site, is crucial. If the active site is not the right shape, it will not bind to its substrate. That becomes important when we talk about enzymes breaking down and denaturation, which we've touched on when we've talked about hyperthermia. If your body temperature gets too high, your enzymes start to break down. The active site fails, meaning that the chemical reaction fails. We'll learn about that more in the next chapter. A specific substrate will only fit the active site of a specific enzyme, and there's those two proposed mechanisms, induced fit or lock and key. So this image shows you both of those. Look closely, pause the video if you need to, but spot the difference. I want you to tell me, which I'm going to ask you in class, I'm going to tell you the answer here in a second, but I want you to tell me which one is lock and key and which one is induced fit. So the one on the right-hand side was the induced fit. You can see the active site is not quite a perfect mirror image of the substrate. But what happens is when the substrate binds to the active site, it triggers a conformational change. In other words, it changes the shape of the active site to wrap around the substrate and hold it in place. And the way I remember this is if you've got a glove laying flat on a table, it's all squished out and flat. If you slide your hand into the glove, it wraps around your fingers. Conformational change occurs with the glove. It's an induced fit of your hand into the glove. Same thing for an enzyme and its substrate. As the substrate slides in, it's like the glove wrapping around the fingers. Don't need to go back to spot the difference. We're not going to worry about that. I should say what I should actually say before I move on from this slide. So this is the lock and key. You can see the shape matches, but this is widely considered not to be the primary way that the enzyme substrate reactions occur. Induced fit is the model. So induced fit is the model that you need to really know in a lot of detail, because that's, if you're going to be asked a question about it, that is the current accepted model for enzyme substrate interactions. So point 10 from that figure 2 in your textbook mentioned putting enzymes above the arrows. This is what I mean by that. So this is actually a catabolic reaction because it's breaking down hydrogen peroxide into its constituent components, water, and also releasing oxygen. So it's catabolic because it's breaking things down. It is probably not entirely coincidence that catalase, being a catabolic enzyme, catalase is the enzyme that actually drives or catalyzes that particular reaction. So catalase will take hydrogen peroxide, break it down, releasing water and oxygen bubbles. Now, when we talk about chemical reactions, they have something called activation energy. So I'm going to illustrate this with a couple of GIFs here. If you've not watched the movie Castaway, you've got very little idea why Tom Hanks is on an island rubbing a stick against timber and talking to a volleyball. If you haven't, you should probably watch it. It's like won Academy Awards and stuff. But what he's aiming to do here is trying to start a fire. He's trapped on a desert island. He has access to no materials, including matches. So he's trying to start a fire so he can actually cook fish and survive while he's stranded and marooned. But as you can see, like, he's got the stick, he's creating friction, it's generating heat. But if you actually watch the movie too, it takes him a really long time to actually get the fire going. In order to get any chemical reaction going, there's a certain amount of energy that needs to go into the system to start the chemical reaction. That's true of fire. He is generating heat. Heat is an energy source, but he's not generating enough heat to actually start the combustion chemical reaction. Fire is a chemical reaction. It's a combustion reaction. But there's not enough energy being generated there to actually start a combustion chemical reaction. So how could you fix that? Well, it could be like this guy here, just add petrol. In this case, petrol would be a catalyst. It reduces the amount of energy required to start ignition. Now, in fairness, he already had a fire going, so he's cheating anyway, but he did start another one. And the way he's done that is by reducing the amount of energy that is required to trigger off that combustion chemical reaction. And that is what enzymes do. They reduce the amount of energy that needs to go in before the chemical reaction begins. So this is an example of that. So if you have a look at these two lines, the blue line is the amount of energy. So this axis here is the amount of energy. You can see a lot more energy needs to go in before the reaction starts. And you've got reactants here waiting to do their thing. Nothing happens. You need all this energy right up the top before you actually start getting the chemical reaction and then products at the end. All right, so that's the reaction without enzymes. If you add enzymes, the activation energy is significantly lower. If you need less energy for the reaction to occur, it will occur quicker because not as much energy is needed to be put into the system first. In other words, enzymes speed up chemical reactions by reducing the activation energy required to start the chemical reaction. If you relate it back to our Tom Hanks and random fire pit guy, so the blue line is really our reaction without petrol. Need to add a lot more energy by adding friction. It's going to take a lot of physical energy from Tom Hanks to start the fire. If only he had petrol, he could have followed the red line and he would have had fire a lot sooner. All right, the from your textbook to show you these examples and also to bring in those terms catabolic and anabolic. It's basically the same graph, but they've just split it in two. Catabolic reaction is breaking down a substrate into two or more products. All right, so your single product comes in, highlighted here in the green. If you don't have the enzyme, you need a lot of energy before you get your two separate products. Catabolic, breaking down into two separate products. And then on this side, much less activation energy here is required to take that green thing and break it into the two constituent parts over here. Anabolic is taking two separate parts and sticking them together to make a new product. All right, so anabolic is building, catabolic is breaking down. And I'll touch on an easy way to remember that shortly. And so anabolic, same principle though, you need a lot of energy before the reaction will occur without the enzyme, but if you add the enzyme, you don't need anywhere near as much energy before it builds that new product. So catabolism versus anabolism. So I already mentioned that enzyme catalyzed reactions can build up or break down compounds in metabolic pathways. So anabolism or anabolic reactions build things up. So an easy way to remember, if you've ever heard the term anabolic steroids, which are illegal and should not be used in bodybuilding, but often are, they use those because it builds more muscle. Now, I mentioned Zach will probably know these because it's talked about in gym. It's not because I'm suggesting anything to do with Zach and anabolic steroids, but they often talk about with respect to getting enough protein and working out that if you don't get enough protein, you start to break down your muscle, which is a catabolic reaction. You're breaking down your muscle. What you want if you're trying to bodybuild is to get plenty of protein. So when you work out, you create an anabolic response to the exercise and you actually add muscle, which you can do naturally and you do not need steroids. But an easy way to remember it is anabolic steroids. Again, not suggesting for a second that Zach has anything to do with that whatsoever, but he's probably heard the terms before. So anabolic reactions are a series of cellular reactions that create molecules from simple building blocks. In other words, it takes simple molecules, sticks them together to make something more complex. And catabolic is just the opposite of that. So they release energy, which is important. When you break bonds, you release energy. So if you break down molecules, like breaking down glucose into pyruvate, and then on it goes through cellular respiration. If you break them down, you know it releases ATP and it releases heat. We know that because we're warm-blooded, all right? And that's the whole reason we're warm-blooded, is because it's releasing heat. So catabolic reactions break things down and they release energy, often in the form of heat. Last point for this chapter, biochemical pathways, which is, there's a figure 6 in your textbook on page 131. I don't really like it. So I've used a different image here that I think explains what we're trying to tell you better. A biochemical pathway basically is a series of chemical reactions where the substrate from the first reaction breaks down and forms products. The product from that first reaction becomes the substrate in the next reaction. So here is your first substrate, this green triangle. It goes into the Pac-Man, hence the background, right? But it goes into the Pac-Man. It breaks it down to form its substrate, its product. But its product is the substrate that then goes into enzyme number 2 in the chemical pathway. It makes its product, which is this blue thing, and that is the substrate for enzyme 3, and then finally you make an end product. So firstly, a biochemical pathway is a series of chemical reactions driven by enzymes. And the product of one enzyme becomes the substrate of the next. The product of the next enzyme becomes the substrate of the last. And hopefully that makes sense. It does show you that in that figure 6, but I find that it's not as easy to follow. This is a simple representation of what we're trying to teach you at this stage. You'll see examples of this when we look at respiration and photosynthesis. So some of the enzyme pathways that go on all the time in your body include growth, repair, respiration, reproduction, digestion, breathing, homeostasis, and there's a lot of immune pathways that are biochemical pathways as well. We'll learn about immunity in Unit 4. So again, just to recap, hopefully now you can, and when I ask you in class, because I'm going to have this presentation, but with a whole bunch of the information taken out, you can define an enzyme for me and tell me what it actually does. So you can also explain its general structure. You can talk to me about its active site. You can talk to me about the way the active site binds with substrates. You can talk to me about the function of enzymes, what they do, and why their shape and those various structures like the active site are so important. As always, if there's questions, I hope to hear from you so I can help answer those. Otherwise, I look forward to constructing a combined and communal understanding of enzymes when we review this together in class. Thank you very much for watching.